Welcome everyone, and thank you for joining us for today's webinar, Clinical Investigations According to the Regulation EU 2017-745. Before we start, I would like to give you a short introduction to the company and the presenters. So NSF International is dedicated to being the leading global provider of public health and safety-based risk management solutions, while serving the interests of all stakeholders, namely the public, the business community, and government agencies. NSF provides services in over 175 countries, with 57 offices and laboratory locations all around the world. The main areas where we provide services are food, water, consumer products, pharma, biotech, medical devices, and management systems. MREX Clinical Research joined NSF International by the end of last year and is located in Germantown, USA and Taiwan. MREX was established as a global full-service contract research organization and is providing comprehensive services in project management, regulatory affairs, FDA application submissions, compliance audits, adaptive study designs, statistical analysis, and general consulting. NSF ProSystem provides global consulting services regarding the whole product life cycle of a medical device and is located in Hamburg, Germany. As members of various standardization groups, NSF ProSystem is actively involved in developing standards such as ISO 13485, ISO 14971, IEC 62366-1, and others. So we're influencing normative policy decisions. Kazempour is the president and CEO of MREX Clinical Research, now an NSF International company. He has over 30 years experience in biomedical research and also prior experience at the FDA and big pharma companies. He is a professor in statistics at US universities and was involved in the conduct of over 100 clinical investigational trials. My name is Sandra Bugler, and I'm the managing consultant for clinical affairs at NSF ProSystem. I have several years of professional experience in the field of clinical and regulatory affairs for medical devices, and I have established a team of clinical affairs experts at NSF ProSystem. My professional focus is on clinical data, especially on clinical evaluations, and as well as on clinical investigations. For the last years, I've implemented processes and templates regarding the new clinical requirements for different companies with different kind of medical devices from class 1 to class 3. Now I would like to start with the content part. So before going deeply into the clinical investigation part, I would like to give you an overview about the current requirements regarding clinical data in general. Each medical device from class 1 to 3 needs to be evaluated regarding the clinical safety, performance, and clinical benefit. A clinical evaluation serves as evidence and is a systematic and planned process to continuously generate, collect, analyze, and assess clinical data. So clinical data could be sourced from 3 different data types. The source with the highest evidence would be the clinical investigation of the device under evaluation. But you could also collect clinical data from literature reviews of the device under evaluation or its equivalent device. In the post-market phase, also clinical experience data should be considered, and this could comprise sales data, complaints, or any other feedback from the market. So that's why there are 2 main clinical evaluation routes. The one we will talk about today is the clinical investigation route, which evaluates clinical data derived from a clinical investigation of the device under evaluation and an additional literature search, which could be available. The second route, which is the most used one, is the equivalence route. In this case, only clinical data of an equivalent device will be considered to prove safety and performance of the device under evaluation. No clinical investigation of the device under evaluation will be needed. But why this approach can in the most cases not be used anymore, I would like to highlight in my next slides. Both the regulation and the METF 271 revision 4 introduce criteria to use for the demonstration of equivalence. Technical, biological, and clinical characteristics should be considered, which are named here. And to prove the equivalence, all those 3 characteristics need to be fulfilled. it is not allowed anymore to take different equivalent devices for different features, as it was done in the last years. The biological and clinical characteristics, such as the same clinical condition or purpose, same materials or substances in contact with the same human tissue or body fluids, should be the same between the device under evaluation and the equivalent device, whereas the technical characteristics should be similar. So the question is, what does similar mean? So there's also a definition which was defined, or which is written by the regulation, and the characteristics shall be similar to the extent that there would be no clinically significant difference in the safety and clinical performance of the device. So let's discuss this requirement on an abstract sample. You would have your device under evaluation and, for example, a predecessor. In the most cases, the predecessor will be the most possible equivalent device, but it will not be identical to your device under evaluation. If you do not have any clinical significant differences, you can use the device as an equivalent device. If you developed a new clinically relevant feature, which is not covered by the predecessor, and in the most cases there is a new clinical relevant feature, that means not all characteristics are fulfilled, and you cannot use the device as an equivalent device. Now, if we look for other possible equivalent devices, you could also look for other manufacturers and other devices. So the new feature might be covered by another device by a different manufacturer, but you cannot clearly demonstrate that you have sufficient level of access to the data related to the devices from the other manufacturer. So therefore, you cannot use the device as an equivalent device. You cannot assess if there are technically relevant or clinically relevant differences for the technical characteristics. And then there is additionally the Section 5 of the Article 61 of the regulation, which describes an exception of the conduction of clinical investigations. However, in this case, the equivalent device is from a different manufacturer, which we just discussed. So the regulation requires then that two manufacturers have a contract in place that explicitly allows the manufacturer of the second device full access to the technical documentation. So that means that two competitors would allow access to their technical documentation to help each other getting the CE mark. And so far, I did not hear from any manufacturer who is really considering this. And as a result, for Class 3 and implantable devices with no equivalent device from the same manufacturer, it is almost impossible to avoid a clinical investigation. And there's always a question if this section would also be applicable to all other medical devices. And if yes, we would need to conduct clinical investigations even for Class 1 medical devices. And if the section is not applying to all medical devices, it is still really difficult to demonstrate equivalence to another device. As a result, for the most medical devices, we cannot use the equivalence route. And we need to evaluate the clinical data of the device under evaluation. This leads always to the question, what would be sufficient clinical data? And this question is really difficult to answer since there is no definition. But mainly, you need publication or somehow reports which are naming your medical device in the context of clinical data. If you did not identify any data within your first literature search, you could broaden your search by revising the search terms or consider other sources such as literature databases, guidelines, technical documents. But however, if you cannot identify any relevant clinical data for your device under evaluation, a clinical investigation needs to be planned. And within the life cycle of a medical device, a clinical investigation needs to be planned in an early stage. The clinical evaluation plan needs to conclude on the clinical development strategy, and before the CE mark, the clinical investigation needs to be finalized. And as said in the beginning, the results need to be included in the clinical evaluation report, which will be part of the technical documentation to prove the clinical safety, performance, and clinical benefit of the medical device. So let me give you an overview of the new requirements regarding clinical investigations under the regulation. In comparison, the Medical Device Directive 9342 EEC has way less requirements regarding clinical investigations than the regulation 2017-745. This is already showing how important clinical data for the current medical device industry became, and is enabling the same standards in Europe. In the following two slides, I want to highlight the general content of the regulation regarding clinical investigations. Besides general requirements, the protection of vulnerable groups, and especially breastfeeding women, pregnant women, minors, are of main importance. So addressing those groups and also emerging clinical investigation and emergency situations are showing how important the protection of the subjects are. The next articles are summarizing requirements regarding damage compensation, the application, the assessment by member states, and the conduct. As for the whole regulation, also for the clinical investigation part, the electronic system is considered in Article 73, and other articles are referring to the conduct of the studies. For example, the substantial modifications, corrective measures, temporary hold or early termination, as well as recording and reporting of adverse events. And there is a new coordinated assessment procedure for clinical investigations in Europe. So but I will get back to this later. Since there are a lot of new articles, there are also new definitions. However, these definitions are not really new. A similar wording can also be found in the ISO 14155 standard for clinical investigation of medical devices. Also, the changed definitions are not completely new. A clinical investigation shall establish and verify the intended performance and the clinical safety of a medical device. And, what is new, also the clinical benefit of a medical device. The Medical Device Directive did not focus on the benefit at all. So now it is not sufficient to only show that the medical device is working as intended, but also identifying a benefit and proving it with clinical data. And as we've seen, the definition of a clinical evaluation includes the benefit as well. So when planning a clinical investigation, one of the first questions the project management will ask is regarding the application and its timelines. In general, the timelines are currently different in each member state. In the future, it will be easier to plan a clinical investigation in more than one country in Europe. And however, the requirements regarding the ethics committee will still be different. So that's still a problem. But for the application for the authority, the timelines are clearly stated, and here you can see the different deadlines. So 10 days after the submission, there is a formal examination, whether the clinical investigation is in scope and/or complete. So this is the validation of the application. If the application was incomplete, the sponsor can submit a statement or other documents within 10 days. The member state could extend this period by a maximum of 20 days when appropriate and applicable. Afterwards, the member state shall notify the sponsor within 5 days of receipt of the comments or of the requested additional information, whether the clinical investigation is considered as falling within the scope of the regulation and the application is complete. The member state can also extend this period by further 5 days. So, but when can a sponsor start with a clinical investigation? I think that's the main question. In the case of an investigational device class 1, or in the case of non-invasive class 2A and class 2B devices, the sponsor can start immediately after the validation date of the application. So that means 10 days after the submission of the application, with the possibility of extending the respective period by 5 days. Additionally, the ethics committee need to approve the clinical investigation. In case of all other investigational devices, The sponsor can start as soon as the member state has notified the sponsor of its authorization within 45 days of the validation date. The member state may extend this period as well by a further 20 days for the purpose of consulting with experts. And also here, additionally, the ethics committee needs to approve the clinical investigation. And as mentioned before, we have also this Article 78 regarding the coordinated assessment procedure for clinical investigations. So for clinical investigation in more than one member state, this article applies. When the electronic system is in place, the application will be submitted once and then forwarded to all member states concerned. One member state will be then the coordinating member state, and the sponsor can also propose one. The completeness of all documents will be assessed separately by each member state, so they are all still involved. Then each member state can make comments which will be included in the draft assessment report. This report will be also reviewed by all member states. And the coordinating member state will then prepare the final assessment report within 45 days from the validation report and notify the sponsor about the decision within 5 days. So again, you have the 45 days as also written in Article 70 about the application for authorization of a clinical investigation. So I would like to summarize the new requirements of the Regulation 2017-745 on clinical investigations instead of listing all articles. So mainly, it can be concluded that the most articles are based on already existing requirements of the ISO standard 14155. Even though the national laws of certain countries were not that strict, the requirements are not new. In Germany, for example, most requirements were even stated in the national law. Only the timelines are in most cases new. In the case of Germany, they are mostly shorter, but could be extended by member states if necessary. And as highlighted in the structure of the articles of the regulation, the protection of vulnerable groups, and especially within the informed consent form, is of high priority. I also talked about the new focus of the clinical benefit, which need to be proven by clinical data. And there's an electronic system needed, which shall be also used as for other new requirements of the regulation. Furthermore, I've talked about the assessment procedure within several member states, which will make it easier to conduct multi-center studies all over Europe. But still, finally, the Commission has to prepare for clinical investigations. So the electronic system needs to be finalized, and also guidelines need to be prepared. So before I hand over to Kazem, I would like to highlight the necessary documents needed for planning, conducting, and closing of a clinical investigation. For the application of a clinical investigation plan, an investigator's brochure, a template for the informed consent, and insurance for the subjects is needed. Before starting, the clinical investigation needs to be approved by the ethics committee and the competent authority. And additionally, if not included in the clinical investigation plan, a monitoring plan and information about the data management should be provided. During the execution, also monitoring reports, test forms, interim reports, or documents regarding changes are needed. So this is all just an abstract of documents. So for closing, for example, the ethics committee and the competent authority needs to be informed, and final reports should be prepared.
Thanks, Sandra, for a beautiful explanation and detailed explanation of the regulation. Let me start by telling you about our experience in clinical investigation in general, and more specific, I will tell you, in medical device indication. In the last 20 years as ANNEX, we have conducted more than 300 clinical investigations. From medical device point of view, we have done more than 70 in Class 1, Class 2, and Class 3. To be more specific, in Class 2 and Class 3 that most often require clinical investigation, we have done more than 50. Medical device trials with EU investigational sites. We have done 8. And let me tell you more about the indications, because not only medical device class 2 and 3, but indication, we have done quite large number in different indications, starting from antiseptics, cardiovascular, diabetes, till wound healing, diabetic foot ulcer, and urology, and very, very diverse indications. From the capability point of view, because not every factories or biotech want all the services that Amarex Clinical Research offers. We offer from the concept, from the study design till setup of the trial, and we do the monitoring as well as finishing the clinical study reports. So we have all the capabilities, but not all the services will be required by all biotech. Sometimes we have clients that they do have some portion of these services or these departments in their own organization. We try to help them to the pieces that they don't know or they need help. We start and helping them. Those services includes project management, medical writing, statistics and biostatistics, data management, from many aspects of it, from electronic data capture to paper to ePro, electronic patient reporting outcome, to WebView, which basically is CTMS suite sites that give you, from the randomization point of view, to IP management and all the related aspects of the clinical investigation. Let me tell you about the compliance, because to conducting clinical investigations, you need to pay attention to compliance. Amarex has been audited by Food and Drug Administration 3 times, and these audits have not been for cause audit. These audits have been for medical devices that we finished the clinical investigations and ready, or were submitted to the FDA, and FDA came and audited us for these medical devices that we submitted the applications. Once was in 2017, and this was for a fairly large 900 subject trials in more than 60 international sites. And it was one of those studies that there were fairly large number of deaths in the study because of the nature of the clinical trial. And the FDA came and audited Amarex with absolutely no finding, or as here we say, no 483, no issues whatsoever. The same it has been in 2015 and 2010, absolutely no findings and no 483. In addition to these audits, we, Amarex ourselves, after a trial is finished and a clinical investigation is finished, we go and select those investigational sites and audit them to prepare them for a potential audit. We choose those sites based on the potential for the sites being selected for audit by FDA, because the regulators go and audit some sites, and the selection of sites are based on some criteria that we have developed, and we use those criteria to select the sites that need to be audited. Okay, said enough about our background. Let me talk about the acceptable data analysis. Sandra mentioned about what type of data are necessary for submissions. Now I'm focusing on the acceptable data analysis for the clinical investigation. Clinical data can be, as Sandra mentioned earlier, can be obtained via many different routes. And I focus on the analysis. Analysis can be retrospective analyses. That is that the main focus is that we need to show, when we are doing data analysis, show that there is no bias there. Retrospective analysis or reanalysis of existing data, or conducting meta-analyses, or looking at a large population and then select some subgroup or subpopulation analysis. These are retrospective analyses. They are not as credible and valid as if you do a prospective analysis. And if in case you are going to do this, you have to talk to regulators in advance, develop the statistical analysis plan and methodology that you are going to use in advance, and see if they do accept that. But retrospective, and let me focus on the prospective analysis. The prospective analyses can be doing a single arm open label trial or randomized controlled trial that may be open, but it's randomized controlled trial, or using the gold standard, double-blind randomized controlled trial. We have conducted all different aspects, starting from retrospective analysis and meta-analysis till doing multi-site, multi-country, double-blind randomized trial, which is the gold standard. Another aspect I like to just mention here, and then I will elaborate on it later, and that is called adaptive clinical trial design. The question that I have here is that can we use adaptive clinical trial design? The answer is yes. Post 2010, the FDA came up with a regulation that allowed to use adaptive trial design. I will explain what we are adapting and what we can modify in adaptive clinical trial design later on. Let me focus on prospective clinical investigation. To obtain acceptable and clinically valid data, we need to start from a clinical investigation plan or as protocol. And also study design has to be detailed in that with evidence of its scientific robustness and validity and rationale for it. Also, we need to focus in the clinical investigation plan about the approach and statistical methodology that is going to be used, especially for the main objective, as we may call it, primary endpoints and secondary endpoints. Also, you need to have an adequate statistical power and for the number of observations that you are going to use in that trial. And obviously, the clinical sites that you are going to use to generate the data based on the clinical investigation plan, those have to be very valid and acceptable sites. Because from regulatory point of view, the PI, the primary investigator of the site, needs to be qualified to do the investigation for you. The endpoints, as I mentioned earlier, are the key components of it, and the right endpoints has to be very clearly specified in the prospective investigational protocol. And the intended purpose, the clinical benefit of it, the performance, and the safety of the device all have to be detailed and need to be how you are going to measure them. Prospective clinical investigations, I'm going to continue on this one because this is really the aspects of it is important. The target patient population has to be very detailed in it. And also the statistical section of that has to be present to say what type of hypotheses you have and how you are going to evaluate that hypothesis. And if the sample size that you have chosen is based on some power calculation and it can be justified. As I said earlier, the methodology that you are going to use, especially for the primary endpoints or primary and secondary hypotheses, has to be detailed. If you are going to use a t-test or an odds of variance, you have to mention it, the methodology that you are going to use. If for one reason or another you think that some population may need to be some adjustment due to some demographic, age, race, gender, or some other factor that you are going to use an odds of covariance, you need to specify in advance that you are going to do some adjustment due to those factors. And analyses that are related to selected endpoints has to be in more detail, especially if you are going to do subgroup analysis. You are going to look at patients by age or by gender. You have to specify that one in advance. Dealing with missing data has to be presented there, regardless of any way that you conduct your trial. Always there are possibility that some subjects are missing or withdraw or lost to follow up. In the clinical investigation plan or in the protocol, it should be specifically stated how you are going to deal with those missing data and how you are going to deal with withdrawal subjects or loss to follow up. I'm going to talk about the sample size and the statistical power because after the endpoint, the statistical power is very essential in any clinical investigation after the endpoint. The endpoint is basically how you are measuring your primary objective of your clinical investigation. Sample size and power really are two sides of one coin. That is that the higher the statistical power, the higher the sample size you need, and vice versa. If you do not have enough sample size, number of observations in your trial, the statistical power will be not sufficient. That means it may not show the clinical significance and statistical significance of the product. The guidelines states very clearly that an adequate number of observations to guarantee the scientific validity of the conclusion is needed in any clinical investigation. And the statistical consideration and justification should be present for that adequate sample size that you are going to use. Generally, it is known that you need minimum of 80% power. You can go 90% power, but you will need a lot more observations for that. But minimum 80% power is required for a sample size for a clinical investigation. And the alpha, or some people refer to it as type 1 error, or the criteria that you choose, is it statistically significant or not, is usually is used 0.05, that alpha 0.05. If it is less than 0.05, we will say is statistically significant. If it is not, we'll say it's not. That people refer to it as type 1 errors or alpha. Why is it important, sample size? Because really cost and time to completion of any investigational trial depends on sample size. So you need to really specify that and make sure that has sufficient power and can really do what you are thinking of the hypothesis to test. This one can test it. Okay, let me tell you about adaptive clinical trial design, because now that I talk about sample size and time to completion and the power, adaptive clinical trial design for the clinical investigation, it is a methodology that 20 years ago, 30 years ago was absolutely not acceptable, because in the middle of the trial, you cannot modify the protocol. You cannot modify the clinical investigation plan in the middle of the trial. But as I said, in 2010, some guidelines came out that yes, there is a possibility that you can adapt your trial, but that means that in the middle of trial, do an interim analysis, look at the result, and say, oh, I thought that I have sufficient power based on 300 observations. You realize that that is not correct, and you need to increase it to 400 or 350. In the past, it happened that when the trial was over, the p-value, rather than being less than 0.05, it became 0.06. And the main reason was they needed 30 more observations, but the power that they selected at the time was not correct. But now using adaptive trial design allows you to use this methodology, and if it needs to be increased, you can increase it. The FDA guidelines came out, as I said, first in 2010 and revised in 2018. EMA also has its own rules, but is it acceptable? The answer is yes. There are other guidelines that need to be used, but it is a methodology that it's very, very strongly recommended. Lately, I would say in last 8-9 years, almost all clinical investigation trial that we do, we recommend to the sponsors to conduct and use adaptive clinical trials. What is adaptive clinical design? As I mentioned earlier, it is a methodology that you will put it in the clinical investigation plan in advance and saying that the trial needs 400 observations based on the statistical power, based on the past history. And then you will explain that after 200 observations or after 150 observations, you are going to do an interim analysis and evaluate both from the safety and effectiveness point of view and recalculate your samples and to see if you have sufficient power. If not, then you will put together a methodology in advance, how you are going to modify it and how you are going to adjust the sample size based on the data that you obtain from the trial. Because usually power calculation is based on historical data, but we don't know that from the time that historical data was collected till now, some other methodology came in market, some other devices are in the market, some other clinical practice are in use that power it's not necessarily correct. The question I have here, can we use adaptive trial design for prospective and retrospective clinical investigation? The answer is yes. In prospective, absolutely yes. In retrospective, you need to talk to regulators and make sure that you are presenting to them why you think by using adaptive trial design, you are not introducing any bias. Will it impact type 1 error rate? The answer is there are methodology to make sure that no, you do not need to pay any quote-unquote statistical penalty, and you don't need to adjust your type 1 error. What do I mean by type 1 error rate? Meaning that rather than 0.05 being your borderline, you have to use 0.07 or 0.06. The answer is all this. If you design your trial in advance such a way that take care of these factors, no, you don't. It will not have any impact on type 1 error. The reason I bring this one up because in the past there was some interim analysis that impacted the type 1 error. What are the benefits of adaptive trial design? Really, I cannot count how much benefits you can gain other than the sample size and power. I will tell you in the next slide what other benefits you can get. But it is always it is beneficial to evaluate it and see if you can use that adaptive trial design to adjust your power and also other aspects of your trial. What are the risk and negative aspects of adaptive trial design? If you do not do it in advance, if you do not plan it, and in the middle of the trial, you try to do that, that is a no-no case, and that is risky. So what I'm trying to say that it's adaptive trial design, I don't see any negative in it, but if you don't do it, I do see a lot of negative from many, many aspects of it. So it is always beneficial. If you are going to do an clinical investigation, definitely evaluate situation, see what are the benefits and the risk of using or not using adaptive clinical trial. The question that may come up is that what are the common adaptations? What can you adapt in the middle of the trial? The sample size and power that I mentioned earlier, that is a very, very, very typical one that a lot of sponsors use because, as I mentioned earlier, the cost and the time to completion of a trial depends on the sample size. And obviously, being able to reach the goal of hypothesis that you have, the power is essential. And as I mentioned earlier, the power has to be minimum 80%. Yes, there are some organizations, some pharma that they don't have any problem to increase the power to 90, 95%. rather than having 500 observations, they may need 800 or 1,000 observations. But some organizations do not have that power. Therefore, looking at adaptive trial design to help you in advance is essential. Also, it is possible to have, rather than two arms in the trial, the control and the active arm, you may have three arms. I just imagine of low dose, high dose, and control. So you have more than one arm in the study, more than two arms. Typically, it is two arms. It is control and active. You may have three arms or four arms, but you don't want to carry the low dose or the inactive arm till the end of the trial. So if the study is, I'm throwing in numbers, let's say it's three arms, 600 observations, 200 apiece, and then after 300 observations, 50% of the trial, you evaluate and you find out the low dose is not working or the high dose is too much adverse events, you may want to stop that arm. So from that point on, you can stop that arm and continue only the selected arm plus the control. So that is the way that you can use adaptive trial design by discontinuing the ineffective treatment arm from the trial. Also, there are times, especially in the medical device and even some other areas, we, rather than going for superiority of one product against another clinical product, we go for non-inferiority, or may call it equivalency. In those cases, we do not want to show it is superior. We want to show it is not inferior, non-inferior, or equivalence. In those cases, if you use adaptive trial design, you may be able, after some time during the study, evaluate and see if you see it is superior, you may want to claim the superiority. If you do not do that, it is very, very hard, and to be honest, sometimes even impossible to change superiority to non-inferiority trial or vice versa. But if you design it in advance and present it to the regulators and say that I want to design it as a superiority, however, I will evaluate it and I may change it to non-inferiority. If you are going to change it to non-inferiority, you have to explain what are the margin of non-inferiority and all the criteria that are necessary to difference between superiority and non-inferiority. That's another one that you can use adaptive clinical trial design. Another thing that is often used, adjustment in the randomization. If you have, let's say, two-arm study, control and active, and you do not need to have, out of, say, 400 observations that you don't need to have 200 for control and 200 for active. You may want to have, say, 3-to-1 randomization, that is 300 active, 100 control, or 2-to-1 randomization, or a different ratio of randomization you may want to have. By doing adaptive trial design, again, in advance, you will specify that at the start you are going to do 2-to-1 randomization, 2 active, 1 control, or 3 active, 1 control, or 3 of your medical device, 1 of the control device, and then do an interim analysis, and then you may decide to adjust it, make it 1-to-1, or continue to be 2-to-1, or the way it is. Another aspects of the one that is used quite often in clinical trials and medical device, in any clinical investigation, is combining phase 1 and 2, or phase 2 and 3. The example that I gave you about having low dose and high dose and control, that means that you are not sure of which dose is your marketed dose. In that case, in old days, we did phase 2 trial and identified the dose, and then we went and did phase 3. There are possibilities that one may develop a process that do phase 2/3, and the phase 2 is up to doing an interim analysis, and decide which dose is going to be the one to use, and then modify it and go on. So there are so many different aspects of adaptive trial design that I did not mention here, but really it is one of the characteristics that helps the clinical investigations to be done more efficiently and be done with lower cost and shorter time. Another thing that I put down here, futility analysis. This is one area that really, before even adaptive trial design was present, and that was some sponsors starting doing it, and then after an interim analysis, realized that based on the current power calculation, really they will need, I'm throwing a number, 2,000 observations to do the trial, and therefore they say it's futile. Therefore, there is no need to continue that. We have had situations like that, rather than continue the clinical investigation, and you realize that really the result after 2 years or after 1,000 subjects, 500 subjects, you realize that no, the power or the hypothesis that you wanted to present data for cannot be based on the whatever reason that you were not seeing. So futility analysis is the one that is often used in adaptive trial design. I spend a lot of time on adaptive trial design really because really it is extremely important, and not everybody is recommending it, but regulatory agencies, all of them accept it. So it is beneficial for you to evaluate that and talk about it before you initiate your trial. I want to just give you one example of a case study that we had that was, I believe it started late 2018, and it was an international client that came to us with an IBD product. They received the CE mark with a clinical investigation done in outside Europe, and then they wanted to go to US FDA requesting approval in US, but when they went over there, they discussed with another CRO, and they were with another CRO for a long time, and then when they came to us, we decided to do a gap analysis to see what they have and what they are going to claim. And based on the gap analysis, we had some discussion with them to establishing the regulatory pathway. And after we agreed with that together, we went to FDA and we presented the scenarios and negotiated with the FDA to try to use as much data from outside US, combining them with some from US. And after discussion and presenting the reason that we believe by doing it this way, we preventing any biases, we were allowed to combine data from non-US and US with very limited amounts from US, and take advantage of all the data that we have from non-US. And we submitted that to FDA from the pre-submission meeting that was sometimes in the middle of 2018, I believe it was, till submission. And we conducted investigational trial in US till we got the approval, I believe was January 2019. So it took altogether under 10 months. I know I talked for a long time, just let me give you the summary of take-home messages on the design conduct report of clinical investigation. As I mentioned earlier, to obtain acceptable and reliable clinical data, we need a clinical investigation plan in advance or a protocol, an adequate number of observations, sufficiently powered, and methodology for statistical analysis, especially for the primary and secondary endpoints, has to be specified. And the endpoint and the main hypothesis has to be specified in the protocol in advance. and obviously the target population also should be detailed. And you need a statistical analysis plan developed before the database lock and before unblinding if you are doing a double-blind study. With respect to adaptive trial design, I believe I talked quite a bit, talking about the cost reduction, time reduction, and also being able to adjust for a randomization ratio. But I want to make sure that all this has to be done in advance, and you can talk with them to regulatory agency using them without any statistical penalty. And I stop here, and thank you very much for listening.
Thank you, Kazem. Before closing the webinar, I would like to highlight our offer for a free consultation in April, May, and June regarding a planned clinical investigation if an appointment is arranged in March. So you could directly contact me under sbugler@nsf.org. And we are also planning a training and a consultation in Hamburg with ANNEX, but we will let you know when this is planned given the current situation. So thank you very much for listening, and if you have any questions, please do not hesitate to contact us directly. Thank you.